Cholinergic receptors mechanism of action. So let's look at the mechanism of action at the receptors now. We've talked about the locations and the actions at this receptor points, M1 and M3, M2, NN and NM. But let's look at now, how do these receptors really carry about or go about their actions? How do they carry out the response to the stimulations? So there's something called G proteins present on the surfaces of the membranes where the receptors are located. So some of these receptors are sort of coupled or attached to a G protein. So let me just draw something simple here. So let's assume this is the lipid membrane of the cell. This is lipid membrane of the cell. Then this is where the receptors are. Let me change the color to blue. This is where the receptor points should be. This is the receptors. These receptors are sort of joined to, there's a protein like this that runs like this on the surface of the membrane, has a terminal point here. So the membrane still goes on like this. So this is the membrane. Okay, I think I've explained it to a large extent. So if this is NM receptors, so this is okay, let's start with the M receptor. This is M1 or M3 receptors. M1 or M3 receptors are actually coupled to a GQ protein. So this protein here that kind of joins to the receptors here, it's called GQ protein. So when ACH, the neurotransmitter ACH, binds to these receptors, M1 or M3 receptors, this protein is kind of activated. And activation of this G protein sort of leads to the activation of something, of an enzyme. The enzyme that gets activated by activation of this G protein is called phospholipase. Let me rewrite that. Make it a bit legible. So phospholipase A2. So this is phospholipase C rather. So phospholipase C gets activated, and what this enzyme does is the enzyme cleaves a protein, and the name of a molecule present on the membrane. The name of that molecule is called PIP2. PIP2 stands for phosphoinositol bisphosphate. So this enzyme acts on this molecule here and breaks it down into diacylglycerol and also IP3. IP3 stands for inositol triphosphate. So what this enzyme does, this enzyme acts on this to cleave it. So it cleaves it into this and into this. Now what this does is that, what IP3 does is that IP3 goes to the endoplasmic reticulum. It goes there and causes the release of calcium. So both calcium and DAG come together now to mediate the contraction that occurs as a result of stimulating M1 or M3 of action. Mechanism of action just talks about how these receptors really carry about their action. So we have M1 and M3 here. They are coupled to a GQ protein, and the GQ protein coupling leads to activation of phospholipase C, which helps to cleave PIP2 into DAG and IP3. The IP3 goes to the ER, which further produces calcium, causes the release of calcium, and calcium on the other hand acts to DAG to cause contraction. So this is just a summary of what I explained here. So M1, M3, GQ proteins, the phospholipase C activation leads to PIP2 being cleaved to IP3 and DAG, and IP3 goes to ER to bring out calcium. Calcium and DAG causes the effect of contraction. Now let's check M2 receptors. What do M2 receptors really do? Let me clear this page. So M2 receptors also, they are coupled to a G protein. So if this is the diagram I also drew for the other one, so let me just make it a bit simple here. So this is the receptor M2, and M2 are coupled to a type of protein here. So let me just draw the protein like this. So it's a GI protein this time, and the GI protein actually leads to a deactivation of an enzyme. So it kind of deactivates an enzyme called, there's inactivation of an enzyme called adenyl cyclase. Adenyl cyclase is responsible for converting ATP into cyclic AMP. So if this enzyme is inactivated, there will be decreased amount of cyclic AMP produced. Probably this is what is responsible for the decreased heart rate effect and decreased conduction velocity effect produced by M2 receptors at the level of the SA and the AV node respectively. So this is just how it works. So this is GI coupled, this is GI. So M2 receptors are GI coupled, and they inactivate adenyl cyclase. So ATP can no longer be converted to cyclic AMP, and there's decreased amount of cyclic AMP, producing decreased heart rate and decreased conduction velocity. So that's what we explained here. M2 receptors, GI coupled, inactivates adenyl cyclase. ATP can't be converted again to cyclic AMP, so there's decreased cyclic AMP and mediating relaxation. So the last one is nicotinic receptors, NM and NM. There are no second messengers like this to having second messengers in terms of IP3, cyclic AMP, DAG, and the others. This one simply just activates channels. So once you stimulate NN or NM receptor, because if this is the membrane here now, this is the membrane. There are channels present on this membrane. These channels are pumps. They are sodium, potassium pumps. So when these receptors present on this pump, this is, let me just say this is NM, N now. Let me change that. Let me use another color to explain it. So this is N, this is nicotinic receptors present here. Once they are stimulated, they lead to opening of this channel for ions to flow through, and therefore mediate their action of contraction or causing twitching, as the case may seem, or whatever effect it can cause depending on the location of the receptor. So this explains the mechanism of action of cholinergic receptors. Thank you.